folks yesterday have already talked a lot about our efforts to design vaccines on Eterna. And so I'm just going to talk about a small part of that, which is something that you've probably seen on the Eterna platform, which is these humans versus computers in minimizing p-unpaired. So I want to talk about what exactly is the p-unpaired measure, how did Eterna players versus computers do, and then also what are kind of some next steps in terms of what to think about for computational vaccine design. And so from the start, I just want to say this has been a huge group effort that has involved so many people, and I feel just very fortunate to get to kind of be the steward of the data, but really it's thanks to a huge number of people in the DOS lab, the Barna lab, and then here I've listed the names of all the Eterna players who contributed just specifically in the p-unpaired challenges, but it's thanks to your contributions that the insights that we have were possible. So RNA is an inherently unstable molecule. The reason for this has kind of a cool geometric reason that I just kind of want to point out. So RNA cleavage is more likely to happen when a nucleotide is unpaired. And the reason for that is that for the backbone to be cleaved, here on the left, the backbone has to get into this funky shape where the two nucleotides are pulled apart from each other. And then for this reaction to happen, this base molecule in solution, the electrons have to flow in this inline way for then these electrons here to be kicked off. So in biophysics, this is often called an inline attack because the electrons have to get in line for this to happen. But if the nucleotides here are in a helix, the same backbone is in a shape that this inline attack can't happen. So our hypothesis and our goal is to try to design more base pairing to stop this inline attack conformation from happening. And we know that RNA doesn't just take on one molecule in solution, but it can, structure in solution, but it can take on many. And so we want a measure that averages the amount of unpaired over all of the possible structures that the molecule can take. So here for a small toy mRNA, I'm showing the minimum free energy structure, but I'm coloring every nucleotide by the average amount of time that it's unpaired. And so you can see that loop regions are yellow, so they maybe are unpaired all the time. But then even in stems, maybe some stems are more paired, but then some spend some of their time being unpaired. And for this whole molecule, I can then assign a number that is the average of all these colors. And so I'm going to call that the average unpaired probability for the rest of this talk. And so this is the p-unpaired measure that Eterna players were trying to minimize. And so again, in the context of mRNA design, where there's astronomically many RNA molecules that might code for the same protein, we can have a wide variety of different p-unpaired values. So, this molecule on the left is pretty much totally unpaired. This one on the right is very paired. And so this mRNA on the left is actually what you get from a commercial vendor if you just ask them to optimize the RNA molecule for this. And this one on the right is actually the minimum AUP mRNA that you get for this toy protein, which we can get because it's so short that we can enumerate all of the possible mRNAs. But for anything longer than about this, we can't enumerate them anymore. And so then it becomes an open question, which is how much can we minimize this AUP measure? And so that was the context for which Do Soon and John Nickel launched this open vaccine lightning round. So we picked 4 model mRNAs that are either mRNAs that are studied widely or ones that were designed by either Do Soon or collaborators to actually be viable vaccines against SARS-CoV-2. And so this goal was humans versus computers. And so here I'm going to show you the computer algorithm that Eterna players were up against. Here's the smiling face of your opponent, Christian Cho. This is an extension of another algorithm that Christian came up with. And so it makes use of a Monte Carlo tree search to sample different RNA sequences. So it keeps track, so it stochastically looks at new sequences, but uses a tree structure to keep track of ones that it has already visited. And so then here's just a little movie of the conformations that RiboTree is finding. as it descends down the branches of one of its tree as it gets to the optimal solution. So for this talk, I'm going to just talk about results from one of the 4 challenges, which is the EGFP challenge, but the results for the others are very similar, and they'll all be available to read in a manuscript coming out very soon. So just for starters, I wanted to do some baseline comparisons before we get to the humans or the computers. So if I just go and randomly sample codons to make an mRNA 100 times, this was the lowest AUP value that I could come up with for this, for the EGFP, 0.38. Here on the left, I have a visualization of the MFE structure, again, colored by the unpaired probability for each nucleotide, just to try to visually get a sense of where the loops are and how paired or unpaired they are. If I only sample codons that are rich in Gs or Cs, I don't do that much better. It maybe looks a little bit more stable, but the overall AUP is still just 0.36. So there's all these vendors we can get mRNAs from too. So Dosun went and got sequences from all the vendors. For many of them, they're also stochastic, so you can get many solutions back. And these are the lowest AUP values that we got from those. So for this EGFP, they even got worse from the mRNA vendors. We wanted to select EGFP because researchers at Moderna have also looked at this as a toy system to look at mRNA translation and stability. And we found that their AUP wasn't even any better than the other controls. So, okay, that's our baseline. Now time to look at some other algorithmic versions. So linear design, many of you may be familiar with. It's from Liang Wang's group at Baidu, and it returns an mRNA sequence that it is an optimal solution for the lowest free energy structure. So it uses a dynamic programming method to get this. So here, now we see that the AUP has dropped by a factor of 2. Ribotree, the lowest AUP that I got for this, also dropped by a factor of 2. It actually had the same value for AUP. It had a totally different shape, so that's kind of neat. So Eterna players got a value of the same exact value of 0.21 within the first coronavirus lightning round, where players weren't even asked to particularly stabilize AUP. And then in the lightning challenge of humans versus computers, the lowest AUP solution ended up being a mod of this sequence for the first, from the first round. So we were very excited to see that for all of these methods, linear design, Ribotree, and Eterna player solutions, that we could see this 2-fold decrease in the degradation of these molecules from our conventional design methods. And if we look at what this might mean for how much of these molecules are left, for this length of RNA, after 30 days at room temperature, the conventional methods would have 92% left. After a 2-fold decrease in the degradation, that same number goes to 96% left. And you might say, okay, that's not a big difference. So what is the significance of taking all the time to do this design? Well, if we look at mRNAs that are under consideration to be vaccines, they're much longer. And the longer our molecule is, there's more of those backbone links where the molecule can break. And so under the same conditions for a real RNA vaccine at room temperature, we might only have 25% left. But if we can 2-fold stabilize it, we would then already be at 50% left. And ideally, we would like to be able to have more than that left. So after this first set of lightning rounds, we were asking, can we still see this 2-fold stabilization or more for longer RNAs? But something that you might have also noticed is that this p-unpaired measure, or AUP, takes a long time to compute for longer RNAs. And so it was really slowing down the Eterna browser and playing the game. And so we were wondering if there was a way that we could still help players get low AUPs in some sort of faster way. So we computed a bunch of other metrics and correlated them to this AUP measure. And we found that the number of unpaired nucleotides in the MFE structure had almost perfect correlation with the AUP. And this might also explain why Eterna players in the first round, without being asked to minimize AUP, found low AUP solutions, because if we just kind of look at a structure and think, can we create more structure, we're reducing the number of unpaired nucleotides there. So Dosun and John Nickel went back and launched another round for a vaccine molecule that Dosun had designed that includes the full spike protein. And you may have noticed, so one was in this more tractable sandbox where we're just looking at the number of unpaired nucleotides. But then for those who think that their laptops aren't working hard enough already, he also launched one calculating the full AUP. And so for that larger mRNA, here are structures from all those other control methods. And then we did indeed see that Eterna players could reach twofold, and I think in this case, even threefold stabilization of AUP. And so this is the structure of the solution from Eli Fisker with the lowest AUP for the full spike mRNA. And it was really interesting too, we did not know this before these rounds happened, that we saw similar change in the AUP for all of these lengths of nucleotides for all the rounds. That was really surprising to us. And this, I just told you how there's differences in folding engines, but we saw similar consistent predictions for AUP, whether it was calculated in Vienna or in Contrafold or in Eternafold. So this was really exciting to us. But a question then, so if linear design is also able, or ribotree is also able to achieve these low AUP values, then why do we need Eterna players? Why do we need more methods than just an algorithm to run? And so maybe it is true that we can run an algorithm to get low AUP values. But something that Eterna players were able to do, which totally was not asked for in the p-unpaired challenges, is they came up with a huge variety of structures to achieve that all had low AUP values. So just one way to characterize this is something called a maximum ladder distance that others have used to characterize the structures of viral RNA genomes. And so we see, again, all of these have an AUP of under 0.2, I believe, but they totally span the gamut of what might be possible for this. And there's so much that we don't know about how RNA structure relates to in vitro stability and persistence and translation, that it's really important going forward to just have as many structures as possible. And so it was really neat to see that Eterna players were able to provide such a wide variety of structures. And so going forward, there's still more avenues that we're interested in being able to explore. So one is, can we go even lower? An open question. Another avenue that is also on Eterna, so there's these DEG score metrics, which are kind of getting a more nuanced way of looking at p-unpaired, which is to say that there might be a difference in the degradation rate between nucleotides in a multi-loop or in a hairpin. There's a lot of unknown unknowns in the structure of large RNAs. A place where people have been trying to explore the structure of large RNAs is in considering the genomes of RNA viruses. And so I mentioned earlier that there's this maximum ladder distance. People have hypothesized that it's been related to that viral RNAs need to be branchy in order to bind to these proteins in their capsid and assemble. But there's a lot that we don't know about biology, and there's all these RNA viruses out there that appear in all other ways to be viruses, but they don't have capsids. I was really interested to learn about these because in a way they might be trying to solve the same type of stabilization problem that Eterna players are trying to solve, which is how do we make a molecule that encodes a protein that can exist for as long as possible. So these are also, they're very short. People use bioinformatics to see that they only encode one known protein. So I pulled some of these out and I wanted to see, well, what do their AUP values look like? And if you look at them, these are very recent. They're basically just, you know, people go and sample some pond scum and then see what turns up. And so here I'm showing 3 narnoviruses, and they also seem to have low AUP values and crazy structures of these like very long helices, but that are all very, end up being very compact and circular. And so I challenge you, Eterna players, to find, to see if you can design structures, you know, as crazy like this that exist in nature too. They have another strange point, which is that their reverse complement also encodes a protein, which people don't understand. So if I go down another branch of the narnovirus family, their AUPs are maybe not as low as those other ones, but they have some crazy structures too. And so I have nothing really more to say, but I just wanted to show Eterna players this and hope that, you know, there can continue to be as many crazy structures and interesting hypotheses going forward as possible. And with that, I will end, and I will gladly take questions.
Thank you, Hannah. Thank you. Are there any questions? The chat was very busy, so.
Let me see. I think I need to stop sharing to go back and see it.
Hannah, I tried to answer a lot of the questions in the chat, at least the ones that I could answer. Is there a new lab that has been launched? If Dosun is on the call, maybe you could tell us.
I think Dosun couldn't make it this morning. Right, and so there's several labs coming up to be launched, incorporating DEG-score and Eterna Fold for these same mRNA challenges and other ones.
Okay, it's not quite launched.
I don't know the specifics.
Yeah, it might be later today.
It's not quite launched, yeah.
Okay.
Thanks very much.